All good? All right, thank you. Paul's talking in 55 minutes, so I'll keep it definitely within that. So I didn't quite know what to say, so I thought I would try and just give you an overview of what we do and see if that was of any interest or if there's any overlap with anybody here. So primarily I'm an experimental biologist. We do some theory, but not much. It's mostly experiments. So that's pretty much what I'm going to tell you about today, experimental data in some way, shape or form. And I guess we're interested in trying to test and quantify evolutionary and ecological processes. And so we do that through the vehicle of population biology. We study populations either in the lab, we conduct experimental evolution, or we look at populations and communities out in the real world to try and get a handle on what's going on. So I'm not going to tell you about any of the evolutionary work we do today, any of the experimental evolution. Maybe I can talk about that another day, or maybe not at all. Mostly I'm going to talk about the ecological and population biology stuff that we've been doing. So I like to do experiments, and I learned how to do experiments with this little thing here called Saccharomyces cerevisiae. I used it to do some experimental evolution way back for my PhD and postdoc. Primarily because, one, I was interested in questions around the function and evolution of sexual reproduction. So this little guy is a sexual eukaryote. You can force him to have sex or not. Secondarily, we know a hell of a lot about the genetics and cell biology and molecular biology of this organism, so it seemed a good thing to conduct for experimental evolution. But increasingly, we're using Saccharomyces cerevisiae and its close and not so close relatives to look at more ecological processes. Obviously, we can find it out in the real world, not just in the lab. And so this has become, this organism and fungi in general, we're starting to use to test more ecological patterns. And fungi are also used to do things like make wine, bread and beer, and so there's some ecological overflow into things that might be economically important. And in New Zealand, primarily that's to do with the production of wine, since that's a massive export business for us, and so I can squirrel a few dollars from the government with ostensibly looking at wine, which is also good fun. So for Saccharomyces and fungi in generally, classically they've been studied from a classic microbiological perspective. And the classic microbiology perspective is really reductional, very powerful insights into the biochemistry of the way cells work, into their cell biology, but the corresponding and complementary ecological understanding of these organisms is essentially non-existent. So we're kind of interested in trying to redress this balance if possible. And in some senses, I think certainly for microbial eukaryotes, maybe not quite as true for bacteria, we're still pretty much with our Victorian ancestors, in that we haven't got a clue about the diversity and distribution of these things in nature. We don't even know where they are, let alone what drives their patterns and processes. So we really are back in the dark ages, I would suggest, when it comes to understanding the ecology of certainly microbial eukaryotes, a bit more advanced with bacteria. So some of the obvious questions are still yet to be answered. For example, in terms of biogeography, which we have very well characterized for big things that we can see, for little things that we can't see, we still, as I say, don't really have much of a handle on whether there are any patterns. So when it comes to the study system that we use is eukaryotes, microbial eukaryotes associated with production of wine. So fungi play at least two roles in this system. First of all, they're the things that can cause disease. So they're the obvious fungi that we know in this system. Secondarily, the roles of other fungi in this system are essentially unknown. They're present in these ecosystems, but their ecological function, other than causing disease, uncharacterized. Secondarily, the other reason that you can convince the government to give you some money is that this grape juice is turned into this by fungi, by yeasts. And they don't just convert sugar to ethanol, they also kick out a whole host of other volatile compounds that make wine smell and taste like it does to us. Most of the things in wine are yeast metabolites, they're not from the plant. Thirdly, a bit more of a stretch to the grant proposals is that understanding why wines differ in different places has some other economic imperative, because a lot of the value to do with wine is tied up with where it comes from. And the French have a marketing genius, and they've really narrowed this down to sell their wine by where it comes from and not from what particular variety it was made from. So understanding variance in these things has a number of imperatives. So if we think more generally, various agricultural crops and non-agricultural crops display differential phenotypes, and certainly for agricultural crops where you're planting pretty much the same clone or the same variety.
Sorry,
that's all right. Why do these differ in different places? And classically, this idea is, it's quite simply that the climatic variance and the geographic variance interact with the variance in the plant in some way, and this intersect defines why you get different phenotypes in different regions. But nobody's really classically thrown into the system what role organisms might play in these ecosystems, specifically what role microbes may or may not play in this variance. So the first question is, well, are there any spatial patterns for fungi or not? So we can break this down into two levels. First, we can think about the collections of different species associated with vineyards and the surrounding environments. So this is classic community ecology. And secondarily, we can focus on just one member of this population, one specific species, Saccharomyces cerevisiae, and look at the population biology of this. So that's the two levels that we're going to look at today, community ecology and population genetics. So the first simple-minded experiment was, so the shocking thing was that nobody had done this before. You think how long humans have been making wine and how much of a model organism Saccharomyces and its relatives are. Nobody had done the ecological work to even test whether you get different things in different places or not. It was mind-blowing. So we did the stupid experiment, which was, let's pick 3 different areas, go to replicate vineyards in those areas, isolate what we can find, and test whether we find any community differentiation between these areas or not. So that's all we did. We took replicate samples, brought them back to the lab, stuck them onto agar plates. We get these blobs that appear. I've no idea what those blobs are. And so in order to elucidate what those blobs are, simply...
Why are they different colours?
Different phenotypes, different species. Your guess is as good as mine.
There's only 2 different colours, right?
That's just a sample of soil that we've plated onto an agar plate and let grow.
Different Saccharomyces or something like this?
Yeah, who knows? So to find out what those blobs are, you can isolate them out, grow them up in a pure form, extract some DNA, and then you typically target some area of the genome that you know varies between species, determine its sequence, ask international databases what those sequences are, and put names to those blobs. So my student isolated over 2,000 different colonies from these different places, analysed a specific area, and then when we look at what we get, we see this. This is simply representing the genetic differences and identities between these different types. So what we can see is that there are a bunch of different names around the edge, so these are different species of fungi, and these percentage represents their abundances in these samples. So there's a diversity of things, unsurprisingly. For Duncan, no Saccharomyces. The question really is whether those collections of things differ in different places or not, and so we can do the real undergraduate thing and just table out what species we find. But that's marginally interesting. What's more interesting is whether those particular abundances are any more or less different than we'd expect to see by chance. And so we can do a very simple community ecology test. We can take each one of our communities and compare it to every other community in a particular area, and collapse the differences in species to a single metric, a Bray-Curtis dissimilarity or something. You can do that for all samples within one region, all samples in another region. Then you can ask whether those differences, what those differences are like between regions. This gives you the average within and between region differences. Then you just scramble up the labels many times and ask whether those observed differences are any more or less than you'd expect to see by chance. And lo and behold, perhaps unsurprisingly, the probability of these communities being the same by chance is very low, low enough to reject the fact that there are homogeneous communities across these 3 areas in New Zealand. Staggeringly, that gets into a reasonably good journal, simply because nobody's done it before, not because it's technically proficient in any way.
Excuse me, how far away do you have to go before they start to change significantly? So if you sample here and you sample a foot away, do you get the same community structure? And if you sample here and you sample here again a week later, do you still get the same structure?
So we have some data that we've not yet analysed looking at that. So I do not know yet, but we've done a range of sampling from millimetres through to kilometres, and hopefully that will give us some steer on whether that's at what level, if any, you see that cutoff.
And how about in time?
Unknown. Yeah, unknown. This is just a snapshot. Yeah. Yeah. So that study and every other study looking at pretty much most eukaryotic microbes in the environment have been conducted using a culture-based approach. That is to say, you have some sample of interest, be it from a vineyard or wherever, and in that sample there's some unknown population community of microbes. You take a subsample of that, take it back to the lab, you stick it onto your media of interest, at your temperature of interest, and some blobs on plates grow. Now there's 2 big drawbacks with this. First, there are, what are the underlying population sizes? 10 to the 7, 10 to the 8, 10 to the 9, maybe more. So if your student or your whoever can probably only analyse a couple of hundred colonies from each sample at best, so the sampling error is just beyond belief. Secondarily, it could be that there are some individuals in this community, like red guy, that makes it into your sample, but for whatever reason red guy doesn't grow on your particular choice of media at your temperature in the lab. You just completely miss red guy. Is unculturable. So there's another way forward that microbial ecologists have been using for a little while now. Same sample, same community, same subsample. Here you circumvent the culture-based approach and you simply extract all nucleic acids from your sample of interest to liberate some population of different genomes, or at least genomic fragments. Then you target a diagnostic area of these genomes, so we classically target the divergent domains in the 26S ribosomal repeat. So it'd amplify up these regions from all these diverse genomes, collect these together, and stuff them through a next generation sequencing instrument, 454 or an Illumina. So you can sequence tens of thousands of these molecules to obtain a population of sequences. So this is what we've done. From the very same 2 samples that I just told you about from the first study, we then went to another 2 regions, Marlborough and Central Otago, and had another 6 vineyards with replicate samples. We used an old technology now, it's 454 sequencing, it's pyro sequencing. It's not as good as the current day Illumina technology, has a number of problems with it. However, these can be bioinformatically dealt with to some extent using these cleaning algorithms. So we've obtained a bunch of sequences, we have then cleaned those sequences appropriately, I hope. We've then compared them to some reference database of known fungal sequences, and this allows us to then partition them into bins of genetic identity. And the question is, what level of genetic identity do we make a cutoff to call a species? And that's a hard question to answer because different species will have different degrees of genetic variances within them. However, from some other empirical work, it seems that on average, anything that's 2% or more different will tend to be a different species. So we've chosen to use a 98% homology to define things that fall into the same species.
Excuse me, what about species that are not in your reference database?
Yes, you don't know about
You
them.
just cluster them, you also have them there.
So you can do a parallel pipeline which excludes this step. You just simply bin things into bins of 98% or more homology, and you exclude this. So that would
So
allow...
it's not your case. In your case, basically, all that is less than 2%, close to a known species is discarded.
Yep.
Okay.
So we're underestimating the true diversity of what's there. But at least then we can put names to the things that we get. But who cares about names? But yeah.
And do you do things like remove singletons from the data set?
Yes, indeed. That's it. Anything that's represented here, any species or operational genetic unit that is only represented by a single species, we would conservatively remove in case that was noise. Absolutely.
So how much do you remove by using that database?
About, I'd have to double check, but my recollection is about a third of the data get removed.
Are we using the database to do the multiple sequence alignment?
Yep.
So the organisms that don't, or sequences that don't have homology, the database, how do you align their sequences?
Well, they will be tagged with a low alignment score, and we have the threshold that we would use would be around 70 to 80% homology. Anything that's less than that, out. Yeah. As I mentioned earlier, this 454 technology has a tendency to increase homopolymers. So if there's a run of A's, for example, it will tend to inflate the number of A's that are really there. And there are a number of other issues. To try and convince ourselves that the noise in the process wasn't horrific, we also included in these a number of clonal controls. That is, we grew up populations of a single clone of a single species in the lab, extracted DNA and included it in this process. And fortunately, we recovered a single species from those controls. So at least the noise in the process isn't horrific, you know. So, what do we see? Well, the first thing that we can look at is the degree to which we might miss things based upon the culture-based approach from the two samples that overlap. So from the first study, my student extracted DNA from just over 2,000 colonies. And from this study, we had an equivalent number of 100,000 reads. So we've got about 43 times the sampling effort. However, we're missing about 95% of the community when you compare these two. So, unsurprisingly, the next generation sequencing approaches for microbial eukaryotes are more powerful in terms of what they're able to detect. That's been shown for bacteria, but not for eukaryotes before. Then we can start to table out things. We can look at the number of reads that we find. Then these are the mean number of species that we see in each individual sample unit. And then we can look at the total number of species that we find in any one area and various other things.
Did you get, I may have said, did you get all the culture-based ones that they all show up in the...
Sorry, yes, they did. No, I didn't say that. They did. And more reassuringly, the rank order was correct. So those things that are abundant in the culture-based approach are also abundant in the next generation sequencing approach.
Are you going to show us a rank abundance distribution?
I don't think I am.
You have it, right?
Yes, I do. Okay, definitely.
Well, I guess the reason I was asking was, is there a rare biosphere in these populations?
Yeah, it's a big log normal distribution.
Long
Yeah,
tail,
yeah, yeah, absolutely. Yeah. So first, relatively, extremely simple question is whether species are randomly distributed in space or not. And so we can evaluate this by the classic Whittaker's diversity, where alpha diversity is the number of species you find in any one sample unit, and you can have the mean of that across any one sample unit. And then gamma diversity is the total number of species that you might find within that specific area. So you can ask, for example, how many species you find in a single sampling unit compared to the number in that specific area. And you can do that across different regions. You probably can't see this because it's very small, but these are these numbers. So on average, you're finding about twice as many species in an area than you do in an individual sampling unit. But perhaps more interestingly, you can ask how that differs at different scales. So you can ask how the number of species differs compared to its regional population, compared to the total national diversity. Now, obviously, if these species were truly homogenised across New Zealand, you would expect those two numbers to be approximately equivalent, right? You find everything everywhere, so it doesn't matter where you go, you're finding about the same number of things. However, if they're different, then that's an indication that species abundances are clogged in some way. So if you compare the regional diversity, which is about 1.5 to 2, to this comparison from an individual sampling unit to national diversity, this second number is much greater, 4 or more, indicating that there's a much greater difference between an individual sampling unit to its region than to the national diversity. And of course, you can evaluate that by creating a null distribution. In essence, you can randomly scatter these species across the sampling space in a simulation, and do that many times and ask what's the chance of seeing that difference if things were just truly randomly distributed. And this hierarchical null model test does just that. So this tells us that on average we should expect to see many more species in an individual sampling unit and in each region if things were truly randomized than we actually do with a reasonably low probability. So we can make a reasonably strong statement that species are not randomly distributed across these regions.
You look at the clumps.
Yes.
Is this taking into account abundance or just
No.
the presence absence?
Yep. This is just richness, just presence or absence, not abundance at all. Yeah. So you read my mind. This doesn't take into account abundance. So what we can look at for any particular species, operational genetic unit, are the number of reads that underlie that species to get a feeling of the abundances of these things. So for example, these are two major fungal phyla, the Ascomycota and the Basidiomycota. In terms of number of species, they're about equally as biodiverse, the same number of species. However, when you look at the number of DNA reads underlying this, you can see that the Ascomycota are dominant. There's many more of them. Because for any one read, we can classify it at every level above genus, we can do this at every other level. So for example, you can analyse things at the order level or the class level and everything else in between. Oh yes, and for the people that care about these things, then you can actually look at what these organisms are and they make sense. A lot of them are diseases of grapevines. Once again for Duncan. Or Saccharomyces cerevisiae, the organism that's very good at fermenting, and I guess for Thomas, is ridiculously rare in these communities. We only find it at 1/20,000th of the community.
But you did find it. You did find reads that.
Well, we can only classify to genus level. Saccharomyces. We can use a slightly more sophisticated test, although not that sophisticated, a per-Hanover. So this uses a permutational approach and essentially conducts an Hanover-like analyses on the multivariate data, which are the numbers of different species in different areas. And so what I'm plotting here is the p-values resulting from these, and in parentheses the r-squared, the amount of variance explained by geographic location in these data. And the bottom line is all these p-values are very small, with the exception of these two. So that is to say, no matter what level of biological organisation we look at, and these are abundances, there's a strong signal for differentiation by region. This is just presence-absence data, and at every level below phylum, there's also a strong signal for differentiation by region. So we can make a nice plot of this. And so here we're simply plotting each circle. The size of each circle is the species richness. So Hawke's Bay is the most speciose, West Auckland and Marlborough intermediate, Central Otago the least species rich. And the thickness of these lines tells us how connected the communities are, how similar they are. So the more thick the lines, the more similar communities are. There's a strong signal overall for differentiation if we do the pairwise between all of these. And you probably can't see so much, though. This is a multidimensional scaling plot, and the first axis here explains most of the variance in the data. Here are all the Central Otago replicates. And so Central Otago, unsurprisingly, by eyeballing this as well, stands out as the area with the most, the greatest difference in community composition. Okay. So simple, but not really been looked at before. So next we went on to do a slightly more comprehensive study. We had more sites, but this time we also included a greater number of niches. So all the data I've just showed you were just from ripe fruit on the vine. Now we're looking at soil in vineyards, the bark of vines, as well as ripe fruit. And we've also looked at surrounding native bush to try and get an idea about the connection of communities. So we had a great number of samples. And for each of these samples, we extracted DNA. And we also enriched for Saccharomyces cerevisiae, and we'll get to that in a minute. We're just going to be looking at the communities here. Now, here's a snapshot of the biodiversity of these different communities, so it's pretty clear. You can see that there's a reasonable difference in the number of species that we find. Soil, whether it's from the vineyard or from local surrounding native bush, is the most biodiverse. Bark, then fruit, and fermentation is the least biodiverse. And overall, we recovered about 8,000 different species. Don't worry about that.
Excuse me, is this again using the same approach with matching them to the database and disregarding them?
That's right. So we are yet to analyse these data properly, but what we do see is some, unsurprisingly, a connection between the communities we find in unmanaged ecosystems, native ecosystems, and the managed ecosystems of vineyards in this case. So unsurprisingly, there's an overlap, but obviously we have no idea about, if there's any transfer between them, we have no idea about the direction. We don't know whether there are members of these communities making it into the surrounding native bush or vice versa. Of less interest perhaps, because we've already shown it, but we do show that for each niche independently, and we're just looking within vineyards now, for the bark of vines, for ripe fruit and the soil in vineyards, there's again a strong pattern of geographic differentiation. So these communities differ according to where they come from. And that was in a different year to the first one, so that has some sort of temporal component to it. But the question really under scrutiny here is why? Why do we find different things in different places?
Question on the previous slide. Is it stronger, the diversity on a regional scale or on different niches of the same place?
You're reading my mind again. Excellent. That's exactly what we wanted to know. So the question, so we find different things in different places. That's not biologically unsurprising. The question we're really interested in is why. And there are two obvious ideas about why you might get different things in different places. One, it's stochastic, that microbes can't move themselves, and so there's some limitation to the degree to which they're dispersed. And so you get different communities in different regions quite simply because they're not well mixed due to lack of dispersal. Or two, the other argument goes, well, microbes are very small, they're easily dispersed, their population sizes are massive. You get everything everywhere, and it's natural selection that defines where you find different things in different places. So which of those two might be the stronger? So the classic issue with studies that try to disentangle this is that clearly as you move through space, quite obviously distance increases, but also invariably the environment changes as well. And so if you take a sample, two different sample points, replicate sample points, and you find things are different, it doesn't help you because both the environment and distance have changed. So the two are confounded. So how do you try and tease these two apart? So there have been some clever statistical attempts to do this. Being a simple-minded experimental person, I wondered if we could utilize these managed ecosystems to try and get a handle on this. So to all intents and purposes, a Sauvignon Blanc vineyard in one area of New Zealand is treated pretty much like a Sauvignon Blanc vineyard in another area. Okay, there'll be climatic differences, but for example, you've got the same clone of the same species planted in different places. And so in some way you can control for the effect of distance here. So the simple question is, what seems to have the greater differentiation in communities? You tend to find different things in different places regardless of what niche you've isolated them from? Or do you tend to find different things in different niches regardless of how far apart they are. So is it distance or niche that's the greater descriptor of differences in communities? And we can kind of do this with these managed ecosystems to an extent. So that's what we've analysed. So we've looked at these communities and we've simply asked, we've got two factors here, the region that they came from, or the niche that they came from, and then there's some interaction term, obviously. Now, all three are significant in terms of their effect on community composition. But then we can look at the amount of variance that's explained by each of these factors, and we see that on average, the niche that the community came from explains more of the variance in communities than does the region. So if niche explains about 3 times more variance, we can make a tentative statement that selection might be the relatively stronger driver of community structure than limited dispersal. Nobody's disagreeing. And here's a plot of that. So here we're plotting in dimensional space the differences in communities. Symbols are the different regions, colours the different niches, and it's quite clear they cluster more strongly by niche. So communities are more similar by niche regardless of how far apart they are. Okay, so that's some of this community ecology stuff. Now we're going to look at one member of the species of this community, Saccharomyces cerevisiae, and ask very similar questions but at the population level. So a little while ago we developed some microsatellite loci, some loci that have repeats in them that vary greatly, that allow us to genotype different individuals of Saccharomyces cerevisiae. And our work and some work from a group in the US tended to show that the New Zealand isolates that we've derived from various niches in New Zealand tend to stand out as being different from strains that come from elsewhere. So there's some evidence that New Zealand has a genetically distinct population of this species. So we wanted to unravel this a bit further, and so another PhD student from all these very same samples, she enriched them, because Saccharomyces cerevisiae, as we've just discovered, is very rare. You have to enrich to find it. So she enriched for Saccharomyces cerevisiae from all of these samples. I've already said this. So, just under 4,000 individuals were collected and then were genotyped. And we found that it was reasonably prevalent in the soil of vineyards, and it was there in native soil and fruits, and it was very common in spontaneous ferments, and that's unsurprising. All up, we found just under 400 novel genotypes, and using rarefaction curves, we estimate there's probably around that many different genotypes hanging around in New Zealand. So the first question was, how do these things, how are they dispersed in space? Is there any evidence that you find different subpopulations in different niches? There's some suggestion that perhaps there's different lineages of Saccharomyces cerevisiae that are preferentially adapted to different niches. And so we tested using a variety of different methods to see whether the isolates we found in vineyards and in native bush were genetically homogeneous or they were structured in some way. We could find no evidence by any test at all that within regions there is any genetic demarcation between different niches. So that is to say, the things that you find in the native bush are genetically homogeneous to the things you find in vineyards that then make it into the fermentation of wine. We'll skip this next bit so we don't overrun for Paul. But what about patterns between regions? So you can begin to table out some standard population genetic numbers. So these are the major regions we sampled. Now these numbers of individuals may look very low. And that's because even though we sampled thousands of colonies, if we found the same genotype from the same sample, we had to collapse all of those individuals because that single genotype will probably have clonally expanded in that sample. And so it would be erroneous to count each of those as a different individual. Having said that though, even though these sample numbers look quite small, the number of different genotypes we find is high given them. And there's a relatively high proportion of genotypes that are specific to certain regions. And again, you can use rarefaction curves to estimate the true underlying number of genotypes that are there and the number of alleles. Then it would be tempting to move on and to use standard population genetic theory to calculate a whole bunch of different parameters. And you can do that, but the problem is that these organisms, these eukaryotic microbes, don't really adhere to the models that Fisher and Wright and the others constructed all those years ago for big things. They have a non-standard life cycle. For example, the mitotic and meiotic phases are unlinked, obviously. These things are unicellular, so each individual mitotic cell is different from another, and selection doesn't act on the group as a whole, but it acts on them individually. These things have a very strong tendency to inbreed. They're not at Hardy-Weinberg equilibrium. So if you start using these old equations to try and work out what's going on in your microbial populations, you'll get an answer, but it doesn't really mean a huge amount necessarily. For example, something as simple as trying to calculate the inbreeding coefficient of these things, if you use Wright's inbreeding coefficient, well, is incorrect because the rate at which heterozygosity is lost per generation in fungi that form meiotically recombined spores or gametes that are constrained within a structure like an ascus, as they are here, then the rates of heterozygosity when these things inbreed are different to the rates of loss of heterozygosity in large organisms that produce independent gametes. So, for example, Saccharomyces cerevisiae, heterozygosity is lost at only 2/3 per generation and not a half as it is in larger organisms. So if you rewrite the equations to take into account of that, you can begin to calculate more meaningful numbers as we've done here. And so this is the maximum likelihood estimate for the amount of outcrossing that occurs in these populations. That is to say, each meiotic generation, if a single spore has a choice of either mating with one of its mates in its own ascus, which have come from the same meiotic division as it, which would be inbreeding, or if one of these spores happens to escape and mates with a spore from another ascus, an independent genome, then we can ask how often those two things occur. On an average, it seems that these things mate with each other about 70 to 80% of the time, and about 30 to 20% of the time, these things escape and mate with somebody else. Lastly, I wanted to get a handle on perhaps a more biologically realistic picture of population structure. So most microbial studies might even go as far as to testing whether there's any structure present in the data or not, and they'd stop there. It's not biologically realistic just to say structure is present or not. It would be much more desirable to try and quantify that structure and then also quantify the degree to which subpopulations might also be connected. And so that's what we've attempted to do here. So the first thing to focus on are these dots, which are the different areas that we've sampled from. We can calculate classic FST values for these populations, but as I've said, it's not clear what these numbers really mean because the underlying assumptions of them aren't really met with this organism. But what we have gone on to use is Bayesian methods that infer the number of subpopulations that might be present in your data. So these methods dispense with the a priori knowledge of where these isolates have come from, but they use the data themselves to try and estimate the number of subdivisions that might be present, the number of subpopulations. Now, this suggests that within these data there are about 7 or 8 subpopulations. And then it can assign individuals to each one of these subpopulations. And further, in this particular model, we've allowed admixture to occur. That is to say, hybridization between members from different populations. And so what these funny boxes are actually lots of little lines, and each line represents an individual. Now, if that line is solid in color, it's telling the output of the analysis is suggesting that that individual has all of its ancestry in one of these subpopulations, mostly. That's what these solid blocks of color are. These broken blocks are suggesting that that individual has ancestry in different subpopulations. It's a chimera, it's a hybrid between different subpopulations. Now, classically, when people have used these approaches, they've then visually inspected these outputs and then made a call on whether the observed structure correlates with their factor of interest, usually geographical location. So for some things like humans, that's reasonably obvious and straightforward. When it comes to something like Saccharomyces cerevisiae here, it's not necessarily clear. So we've gone on to develop this statistical tool that allows us to analyze these ancestry profiles in regard to their geographic location to determine whether the differences we see are any greater or less than we would expect to see by chance. And secondarily, which one of these subpopulations might be driving structure more than any others. And so this is something called obstruct. So unsurprisingly, the ancestry profiles of these individuals from these areas does correlate very strongly with where they came from. That is to say, there's reasonable evidence that there are subpopulations residing in each of these different regions, but it's clear and obvious that that's not absolute. Every subpopulation has some individuals that are hybrids between others. That is to say, there's some level of connection between them as well. So we went on to try and work out the degree to which there might be migration between these different regions. You can use classic old estimates of the number of migrants, or there are Bayesian methods that are implemented that allows us to do that, and these Bayesian methods are bidirectional. So these arrows and the size of them is an indication of the amount of movement of these various genotypes from one region to another. Some regions are very poorly connected. They have very small arrows, certainly very small arrows going to them. Maybe coming away, they might be slightly larger. Other regions are very highly connected. And unsurprisingly, this correlates with the influence of population structure as well. So, all up, populations residing in Nelson and Central Otago, which are more satellite regions, have more distinct populations and experience the least amount of migration, inward migration. These regions are more central, and they're the most mixed and most connected. And this region is intermediate. And this pattern is nicely in line with the flow of fruit and paraphernalia and people by the actions of the New Zealand wine industry. So it seems that there's some underlying natural population structure, but that there's been some human influence on this to some degree. Lastly, the last little story. Okay, so Saccharomyces cerevisiae is hanging around in New Zealand in different niches. Why? Why is this species there? Was it there before humans arrived? We must bear in mind that humans have only been in New Zealand about 800 years. Or did humans take it there? Have they artificially expanded the range of this species? So to answer this, we took 52 maximally divergent genotypes using k-means clustering, and we resequenced these on a high-seq instrument, so we have very high quality sequence data with a high amount of coverage. Then we reconstructed a phylogeny and asked where the New Zealand isolates fell. All of our New Zealand isolates fall within this very tightly grouped clade, which all either derive from Europe or are associated with winemaking. And there's this second clade where New Zealand isolates fall, and the two other isolates are from European soil. So all things considered, it's pretty obvious and clear the New Zealand isolates have derived from, share ancestry with, and are in fact part of, a population from Europe. Recently, a massive diversity of Saccharomyces was found from a sampling effort in China. And so this puts the species into perspective here. This is the genetic divergence of the entire species. And here's a little wine European New Zealand group up here. So it seems obvious, it's tempting to conclude, that the short divergence times and tight relationships with the European isolates mean that humans took Saccharomyces to New Zealand, along with perhaps their winemaking paraphernalia. But to try and get a handle on that, well, can we estimate the number of events? Clearly, the upper limit could be very large, not infinite, but it could be very large. It could be happening every day. Every time somebody from Europe comes to New Zealand, they might be bringing Saccharomyces with them. We can't quantify that. So we wondered if we could put a lower bound on this. So we can, if we use a parsimony approach, we can put a lower bound. If there's a clade, so green are isolates from New Zealand, red are isolates from Europe. So we're just looking at the European wine New Zealand clade now. So the most parsimonious explanation for the observation of a clade that only contains New Zealand isolates is that their ancestor seeded this population in New Zealand, and this is radiated since. The most parsimonious explanation. So if we apply that, we can ask what are the minimum number of transfer events from Europe to New Zealand? And the answer is 10. Now we also are able, you can't really date microbial phylogenies very well because there are very few fossils. But there is one tentative date of the split between Saccharomyces cerevisiae and its closest relative. If you take that date and take the lower bound of that very sketchy date, you can then take the lower bound of the nodes in these phylogenies, and here is where they stack up. So these are thousands of years ago. These are the 10 events, 1 being here, 10 being over here. And here is when pretty much humans arrived in New Zealand. Now, given the sketchy confidence limits around these, other than perhaps this clade, it's pretty clear there's no compelling evidence that Saccharomyces cerevisiae was in New Zealand before humans arrived. And indeed, this clade itself is highly likely to be not an artifact necessarily, but we must bear in mind that the New Zealand population has been heavily sampled by us, but the European population, as you can see by the lack of red numbers here, hasn't. If you sampled Europe as intensively, you just need one or few to interdigitate here, and that would increase the number of events and correspondingly decrease our inference of timings. So I reckon that's the most likely explanation. So the bottom line is it's pretty clear that humans have facilitated the trans-hemisphere range expansion of Saccharomyces cerevisiae. And that is all I've got to say.
There's no reason that Saccharomyces couldn't have made it to New Zealand before humans, right? Sampled outside. What about the possibility that European isolates outcompeted in the place? Interesting to know if they're, I mean, given that there's stuff in China, it's from Malaysia.
You're absolutely right. There's no theoretical reason that it couldn't. And we included a relatively intensive sampling effort of all the national parks on North Island. So they were humans, they're not human-free, of course, but they're certainly well away from vineyards. And we only found one Saccharomyces cerevisiae. Found lots of other Saccharomyces.
Right.
And that one Saccharomyces fell squarely in that clade. But yes, in principle, there's no reason that it couldn't, but the data say
I've just
no.
been at a yeast meeting. I know there was some interest in a study that showed that Saccharomyces was using wasp nests. And some other, I guess Drosophila is another one, related bees.
Yes.